Welcome to the Breaking Bio podcast for July 4th, 2012. I'm your host, Stephen Hamblin, for the University of New South Wales, and with me today is Morgan Jackson. Say hi, Morgan.
Hi, everybody.
All right, and all of our various co-hosts, some of whom you've seen before and some of whom you haven't, are off, doing more exciting things. Most of them are at the Evolution Conference coming up next week. That's in your neck of the woods.
It is, up in Ottawa, which is kind of making me bitter that I can't get the 5 hours up the road to get to it when everybody else is coming around the world for it. But it's an expensive conference, so I'm not that bitter.
How expensive is it?
I think it was like $450 for a student registration.
Boy.
That was early, too. It's really, really expensive.
All right. 7 potential co-hosts on this podcast, and so far we've seen 4.
Yep.
All right.
Yep.
And you're the big star with the repeat second appearance.
Well, I won't catch your 3 streak here, but I'll try. I'm also unemployed, which might explain things. I have a lot more free time.
What is your current status?
I work on contracts at the moment, and then I'll be starting my PhD here at the University of Guelph in the fall. So, like I said, I have some free time at the moment.
So who are you going to be working with?
I'm going to be working with Steve Marshall here at the University of Guelph. I did my master's and my undergrad thesis with him, so there's a bit of the whole, ooh, maybe don't do that thing that's going on. But the way I see it in our field, there's very few people, and most of them have all done their work with Steve to begin with. So I don't have an issue with that, because now I'm outreaching and networking with all of you cool peeps.
So what would you call your field?
Insect systematics is generally what I call it. So it's encompassing natural history, taxonomy, phylogenetics, evolution. So we kind of touch bases on all sorts of different things, looking at functional behaviors, functional morphologies, speciation, all the species level aspects of evolution, essentially, above population level. So not much population biology, but more metapopulation, if you want to call that kind of the species concept idea.
I keep hearing, again and again in the literature, and when people are talking over beer, and they're saying, taxonomy is dying. There's no practitioners, there's no money, there's no spirit left in it. I don't know. What? Obviously, that's not my area. What do you think? Is it a dying breed?
It's not so much. Well, it's not so much dying as being left out in the cold to die, like Star Wars. So we're having a lot of trouble getting money. There's some money coming in, like there are some grants in the U.S. that have been quite successful. There's a whole stream of NSF grants called the PEAK grants that are geared for taxonomy. But a lot of independent researchers are having a lot of trouble getting money just to do taxonomy. They usually have to get money to use applied questions or answer other questions, and then they do taxonomy sort of on the side. There's very few labs that do taxonomy, insect taxonomy at least, purely as their research focus. So I'm lucky in that regard. That's where I'm at, where we can concentrate on it full time most of the time. So with very little money coming in, it's hard to attract students, and so that's the other issue, is that there's not a lot of grad students coming in wanting to do taxonomy for various reasons. It's not the sexy science that you see, some molecular biology and stuff like that. And then as well, it's hard to say, come be taxonomist and you'll have an awesome career, because there's very few of them, and so they're coming up very slowly. But I think it's going to turn around in the next couple of years as everybody starts to realize the lack of biodiversity knowledge that we've got, and how we're starting to run ourselves into a bit of a hole without having the right names and the right concepts and the right people to understand them, or to help with them in some regards.
Do you, I know, especially in Canada right now, there's a serious issue with, lack of science funding, and a general political backlash, you might call it, against science and the government. Is that affecting taxonomy in Canada? Is it?
Well, for most of taxonomy's funding, it comes because we're, I wouldn't say on the theoretical side of things, but we're certainly not on the applied pay grades or funding lines. And so we have pretty much the only source of funding we can get generally is through NSERC, which is Canada's National Science and Engineering Research Council, which is kind of like the NSF for Canada. And so it's pretty much the only place we can get funding unless we do these side projects where we can work on groups that perhaps are of agricultural importance, or sometimes you get money if you're working on mosquitoes, black flies, and the like that are of medical importance, so you can get some funding there. But for the majority of species on Earth that have very little impact on human life in a day-to-day capacity, there's not much funding, so we have to go through NSERC. And the problem with NSERC is that they just announced this last year that they're going to try and refocus and go more towards sponsoring more projects and programs that are going to have pretty much a deliverable at the end of it. So they're turning it more into an applied research granting system. So they're going to try and favor projects where they can actually, get a patent out of it at the end, or some sort of technology at the end, which again isn't really going to be very conducive for us taxonomists that are studying tiny flies that are putting names on it. It's important work and important science, obviously, but it's hard to explain it and to get other people who are trying to get a dollar out of it to actually appreciate it at times.
I feel your pain. I mean,
yeah.
PhDs in theoretical behavioral ecology. It's hard to get people to subscribe to the joy and wonder of that. So I know what you mean, but it's sad. I mean, there's so many species out there that we don't know anything about. There's implications for that in our understanding of biology. And, I don't know much about the actual practice of taxonomy, but I do weep for the fact that it's being so underappreciated.
Yeah, so it kind of sucks in that regard. It's a little bit stressful on a day-to-day basis, knowing that it's going to be a bit of an uphill battle. But like I said, I think that things are changing fairly rapidly, and I think that in the near future we should see a turnaround, and I'm hoping to see a turnaround in public attitudes and funding attitudes towards our work soon. So.
Well, we who hope to use your research salute you.
Well, we'll do our best. We'll keep chugging.
All right. Well, from that exciting and somewhat depressing note, we should move on to mention briefly what had to be the world's biggest news story today.
That nobody understood.
Yeah. It's sadly like Lonesome George from last week. Everybody getting on board and trumpeting the brilliant step forward and, getting all excited and having no clue what they're talking about or...
Yeah, the significance or what it even is that it is. I mean, at least with Lonesome George, you can see it, and you can see it's a tortoise, and then you realize that that tortoise is no longer here. You get to the Higgs boson, you can't see it. Nobody can actually see it. They don't actually see the evidence of it. It's all crazy math and chemistry building back to try and trace it back, but nobody really can understand what it does. There's a really good video actually online that I think it's one of the science correspondents for The Guardian in the UK did. It's a really nice article explaining what this Higgs boson is and how it actually imparts mass onto other articles, onto other particles. And it was really good. He had a nice video including ping pong balls and brown sugar, and it made it really easy. Even I could understand sort of what was going on and why it was so difficult to find this thing and why it was so important. So certainly I'm sure that we can put up the link afterwards, and I think everybody should go watch it because it actually was one of the better pieces of science communication I've seen for a very, very complicated topic like this.
Lawrence Krauss had a nice piece on it, actually. It was very, very well done, and a very sober look at it. You know, this is a good advancement. You know, it's great for physics. You know, now we get to go do the fun stuff. $10 billion to find this.
Carving up half the countryside in the middle of Europe.
Yeah, there was actually a note of reproach in his piece because, of course, the U.S. had been building the super colliding, what's the actual name of it? The
Large Hadron Super Collider.
Well, yeah, the LHC is in Europe, but the U.S. had been building the LHC in the U.S. until the mid-'90s when they canceled the project halfway through.
Yeah.
There's a giant hole somewhere still in the southern U.S. I think it's in Texas, isn't it?
Yeah, I think so too.
And he was mentioning this. That there was this note of, well, you know, we could have done this, and we just didn't have the will. And now it's in Europe.
Yeah.
Which,
it's pretty much the moon race all over again. They just didn't get behind it this time.
Well, you know, I mean, they had to lose the space race the first time to go to the moon, didn't they?
Yeah, pretty much, I guess.
But,
so next time they're going to be, all of America would turn into a super collider, and they'll throw everything at it, perhaps. Literally throwing all the particles. Dark matter farm, right there. And then soon we'll be warp driving.
Now you're just making stuff up.
Totally not, man. It's from Star Trek. It's totally going to be real. Yeah, that's real. Somebody tells me that's real.
You're one mention away from being Deepak Chopra here.
Oh, no. No.
Gene
Roddenberry wouldn't lie to me.
I keep waiting for Deepak Chopra to show up and, you know, spout some ridiculous nonsense about quantum mechanics and the LHC now.
Yeah.
You know, how he's going to use the Higgs boson to, you know, cure your arthritis or... I mean, that has to be next, right?
Oh, I assume so. I assume so that these nanoparticles will be injected right through you on a more daily basis than they already are.
If you think about it, there's actually a really natural link. I mean, you know, you've got homeopaths who take things and, you know, dilute them, you know, 1 to 30 million. All they have to do is merge that with the Higgs, and, you know, now they've got super mass natural medicine something or other.
I think we should just patent this idea right now and just make the $1 million off of it. I mean...
Yeah, you know...
Who cares about reputation in the scientific community? I think it's overrated.
The dark side is appealing, you know?
It is. It is.
They have
so much power.
They have money, you know? Like, the one aspect of the Higgs story that I did see that really kind of got my blood going was this this note that the CERN scientists who were presenting the data used Comic Sans in their slides.
Oh, no.
Oh, yeah.
Oh.
Yeah. And dude,
so they could solve the world's greatest mystery at our time, but they can't figure out how to use PowerPoint correctly. There's more than a little irony in that.
$10 billion on this project, and they couldn't afford a better font.
That's,
you know,
that's really sad. Helvetica is like a $25 download. Oh,
I'm pretty sure that CERN scientists today were directly responsible for the deaths of at least 5 designers.
On the plus side, there's now a job opening for at least 5 designers.
I suppose that's true. I like how you look on the bright side. That's about all we have to say for the Higgs, I think. Since
without talking about it at all. Talked about everything around the Higgs. It's like we're one of those little particles circulating around it without actually talking about any of the science.
We briefly talked about our vague understanding of the science.
Yep, I'm good with that.
I'm good with that, too. So let's move on to this story that you put into the lineup today, this the world's smallest fly discovered. So why don't you tell us about that one? All right, so we're back on air here. Had a bit of a technical glitch while Chrome decided to go on vacation. And why don't you just kind of recap some of the amazing biology that was going on there?
All right. So I'm not sure where these edits came in and out. So we've got a fly, tiny little fly, size of a housefly's eye, smaller than the fruit flies that you see flying around your house. So we've got a tiny fly from Thailand, found by a researcher who lives and works in Los Angeles, California, named Brian Brown. This Thailand fly is an antecapitating fly, so it goes about its life. It's a parasite, lays its eggs inside of an ant. The larvae, the maggot of the fly, develops inside the ant, eating the inside of the ant from the inside out. Eventually it makes its way into the head, eats the jaw muscles of the ant, then moves on to the brain of the ant, turns it into literally a mindless slave, mindless zombie. And then when it's ready to develop as an adult, as a human, there we go, we're on the zombie land here. When it's ready to change into an adult, metamorphosize into an adult, it severs the connection, the membrane between the ant's head and the rest of its body. So the head falls off. The larvae of the fly will stay inside the head, and it will use the ant's hard head, as a protective case for it to undergo the last stage of its development. So it'll pupate within the head of the fly, or in the head of the ant, and then later it'll chew its way out of the head as an adult and emerge and start the cycle all over again. And so the fly can only be as big as the head of the ant for which it parasitizes. So you're restrained there. So this is where Brian took this next leap, and he hypothesized which species of ant this fly is probably a parasite in. So he's gone back. He's been working on these flies, these antecapitating flies, for a long time. And so he's gone through and he's actually tracked which species of fly parasitize which species of ants. And he's actually lined up the size of the fly versus the size of the ant and the size of the ant's head. And he's found this awesome correlation where bigger flies are parasites in bigger ants, because there's more room in the head, essentially, and smaller flies are parasitoids of smaller ants, because there's less room in the head, so they have to be smaller to fit into the head. It's like trying to get like an NBA player into your college dorm. It's not going to happen so well. There's a little bit of cramping going on. And so what he's actually been able to do then is he's actually gone through, he's followed this correlation, this graph, and he's hypothesized the size of the ant that this fly is going to be parasitizing. And it's going to be slightly bigger than the fly. So its head capsule is going to have to be slightly bigger than 4 millimeters. And so what he actually found is there is an ant that lives in Thailand in the same sort of area. I don't think they're recorded from the exact same national park, but they will likely be found together in the same area. So he's found an ant that actually is the same size and should be the right size for this fly to actually develop in. And so what he's done is he's taken the functional morphology of this fly. He's figured out through his other work with other species of flies, he's figured out exactly which ant we should be looking at, which species of ant we should be looking at in Thailand to try and find the host for this fly. So there's only a couple of specimens known. It might only be a single specimen known, actually, of this fly, a single female. And yeah, it's only a single female known of this fly. But now he's figured out which kind of ant we should be looking for to find more of these flies. And so he's kind of figured out the biology of this fly from a single specimen based on all of his accumulated knowledge of other flies that are related to it. And so this whole idea of discerning a species' complete biology from a single specimen that he didn't see alive, nobody's seen this thing alive, it was captured in a trap. So it was captured in, they put out a trap in the woods, this fly happened to stumble into it, it ended up in the alcohol solution that we use to catch and kill the insects and preserve them, got sent across the ocean to Brian in Los Angeles, and he went through and found it there. So nobody's seen this thing alive. If any other case, we wouldn't have any idea about its biology other than, holy crap, look at this tiny little fly. What's going on here? But because of his extensive taxonomic knowledge and because he can trace back through the taxonomy, he knows that it's most closely related to another species of ant-decapitating fly, another species in Africa that goes through the same behavior of developing within the head of another ant species. So by combining all this different information, he's able to predict exactly which species, or at least which genus of ant, to look for to find this fly in the wild. And I think that's absolutely fantastic and an amazing story for the value of taxonomy and how it's not just counting and listing species like some people think, but it's actually one of the core evolutionary biology disciplines and can give you a perfect roadmap for how science and how the natural world is unfolding right below our eyes, or even not in front of our eyes. We can't even see this thing. There's an awesome picture on Brian's blog at flyobsession.net of the actual slide, like your microscope slide, would be the actual fly on it, and it's a speck. Like, it's smaller than the type on the typing on the slide. So these flies are super small, gone unnoticed, and he's traced back its entire biology and natural history and the entire food web essentially for this fly from this single specimen and his accumulated knowledge. So that is probably one of the coolest things I've seen in a paper in quite some time. It's pretty exciting. So
that is pretty awesome.
Yeah.
So
yeah, go ahead.
Oh, I'm just kind of bursting with questions. This is a really cool system. Do the ants they decapitate? Do they have any sort of defenses?
Yeah. So we're going to get into super ant time story here. So these ant-decapitating flies are really popular. There's a lot of species of them, and they're all over the world. So some of them are actually parasitic on leafcutter ants. So these are the ants that are in South and Central America. They're the ones that you see on nature documentaries where they're carrying the leaf over their head in the line. So you see all these lines of ants marching. They take the leaves back to their nest, and they feed them to fungus. So they're not actually eating the leaves. They're cultivating a fungus in their nest, and they eat the fungus.
Right.
So what these ants have got is, because they're out in the open for long stretches of time while they're walking back and forth from the plants to their nests, they're very, very susceptible to these flies, which will come in, they'll buzz above these lines, they'll find the lines, they'll buzz above the lines, and they'll zoom down and insert an egg inside of the ant's head. And that one's toast. So these ants, now I don't know whether this is going to be true, I doubt this is true for the other species of this actual tiny ant, but for these Atta ants, these leafcutter ants, they've actually evolved an entire class of sisters, so an entire body form that is specifically meant to protect against these flies. So they're called minims, and they're like really tiny miniature ants that ride on top of the leaves, and they act as little protectors. So they sit on top of the leaves as these ants are carrying them back through the forest. They'll sit on top of the leaf that the ant is carrying back to its nest. So these tiny little ants hitching a ride, and their entire job is to sit on top of that leaf and protect the bigger ant that's carrying it from these flies. So it's like a tiny little pit bull that they're using to protect against these parasitic flies. So you can see this fly parasitism has completely driven an entire new social order in these ants as a method to get away from getting their colonies completely destroyed by these parasitic flies. So like I said, I don't know whether there's anything like that going on in this other group. It's a different genus of ants, and not all ant groups have these sort of structures. As far as I know, it's just the Atta or the leaf-cutting groups that have these extra minims, the tiny little ants that ride. But I assume that there are other protections against these things, whether it's grooming or keeping heads down or not being as exposed outside as often. Which is where this tiny little ant, we can take another look at it, and we can see that it's all small and short, and it doesn't have many protuberances, and it's kind of all compact like a little jelly bean. And so I think Brian actually postulates that it lives within the ant colony itself. So he doesn't have to go out and fly through the forest necessarily to find it. They live within the ant colony, and that way they can actually attack the ants in their home rather than having to wait for them like the Atta ant parasite has to wait to find them in the forest and attack them outside the colony. They're inside the colony, and they're attacking them from within. And so that's why they have all these minute differences. So they look like jelly beans, so it's harder for the ants to pick them up and cause damage to them and pick them apart, because the ants are pretty aggressive that way.
So do these flies, do they show like specialization for this particular species of ant that they're parasitizing, or are they generalists?
I would suspect that they're probably specific. I think a lot of the phorids are very host specific, and I would assume that this fly being so damn small is probably quite specific on this ant. Maybe on 1, 2, 3 species in that area, if there are multiple super small ants like that. But I would suspect that, especially in this case where it's literally the head size has been so correlated, I would suspect that this is a long-established specialization that they're probably dependent on this tiny little ant at this point.
That makes sense. It's like that real estate saying, location, 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 right?
Yeah, it's kind of important, especially if you need that species of ant or whatnot to get through. So, anyway, fascinating paper, short paper, free paper. It's open access as well, and so anybody can go read it and take a look at it. And it was just brilliant in my mind about not only the cool-looking fly that it exposed, but also this intricate and amazing biology that was explained through a single specimen, and our knowledge that Brian has accumulated. It's just cool.
So you mentioned that this was almost an accident, that it was in this trap and they came across it. Is it a common occurrence in insect taxonomy, just stumbling across a new species?
Yeah. So most of our work is done off of these traps. And so what these traps are, they kind of look like a tent. So if you think of like a pup tent, your standard triangular tent with the sides and then they peak up to the top. So what this Malaise trap is, it's called a Malaise trap. It's essentially that, except instead of having outside edges, it's got open edges and a single netting in the middle of it, and then the same tenting roof on top. So as things are flying through the forest, they hit this netting, and generally with flies at least, when they hit something, they'll fly up. And so when they hit this ball, they'll fly up, they get funneled up into the top of this net. That net will go into a bottle of alcohol, they'll fly into the alcohol, and then we can pick them up a couple days later, a couple weeks later. So we use these traps all over the place. We also use other types of traps like pitfall traps, hand traps, and stuff like that. Again, so that we can collect insects, get more insects on our trips than we're putting in time. So instead of collecting them all and seeing them all, we're using these traps to try and increase our productivity. And so a lot of these times, especially with these weird things, you only get a single specimen, maybe 2, maybe 3 specimens at a time. And so a lot of insect species are described off of what we call singletons, or very small series of insects. So if we find something interesting like that, we can go back and more intensively sample an area. So now, say, Brian could go back to this area where he found this fly, to this specific spot that we found it, and he can take a closer look now and look for this unusual fly and try and find it in its location. But for the most part, all of our traps are literally trying to get as many random specimens as we can, so we put them and increase our odds of picking up something interesting by going to new localities or using different trapping methods than we've used before, and then just sorting through all of the random things that happen to stumble into these traps. So it's like the giant, it's essentially a very, very, very random, what's it called? Transect is essentially what it is. So you're transecting the wild with these traps and just hoping that something interesting goes into them. So.
You know, I don't mean to turn this into the interview Morgan hour, but I'm actually really interested in this. Forgive my ignorance. Again, taxonomy, not a strength of mine, but I'm curious. I mean, when you pull these specimens out, I know that you were mentioning, there's a lot of taxonomic knowledge, and you acquire experience and such. But what happens when you come across a particular specimen? Do you tend to recognize them on sight? Do you immediately notice, oh, hey, I've never seen that before? What's the process of discovery?
So it depends. Like I said, a lot of our time is spent going through these samples or going through museum collections. So that's where we get all of our data from, is from either stuff that we've sampled ourselves. So when we go out in the field, we'll have these traps and we'll bring it back to the lab a couple of days later, a couple weeks later, and go through it. We'll also go through as many museum specimens as we can. So we go through like specimens from the Smithsonian Museum in Washington, the National History Museum in London, all the different natural history museums will pull specimens because they've got accumulated material. So we'll generally be looking for things that we're specialists on. So in my case, I could be looking at stilt-legged flies. Brian would be looking for phorids, these anticapitating flies. If I had seen this thing, I probably wouldn't have had any idea what it is. I'd say that's fantastic and cool, and I'm going to pick that aside. I'm not going to do anything. I'm going to send it off to somebody else to work on, but that's pretty cool. When Brian sees it, because of his accumulated knowledge working with these phorids, he's able to put it probably onto the tree of life fairly quickly. And as we become specialists at these projects, this is where taxonomy, again, takes a significant amount of time. It's not something that is as easily picked up as other things. You need to become intimately familiar with what all these different species look like, so you can start placing them. But as you develop that knowledge, and as you come up with this background of knowledge, this roadmap of the tree of life for your group in your head, you can start fitting in things places where they fit. And so Brian likely came through, was sorting through this residue, this trap stuff that was brought over from Thailand, and he probably came across it and found it, and then just worked his way through it. When he saw it, he probably pulled it aside saying, that's pretty unique, that's unusual, I've only ever seen something that looks like this from Africa, which suggests that this is probably something new. So then he'll pull it aside, he'll mount it up on the slide like he showed in his blog, and then he'll go through the process of describing it and putting up the paper. So it really backs on a lot on prior experience, sorting residues, and like I said, understanding what other species have been found before for the group. So
is this something that amateur scientists or interested civilians, as it were, may be getting involved in, or things they can contribute to?
Yeah, well, taxonomy is a massive amateur. I say amateur. By amateur, I mean not paid to do taxonomy. So taxonomists, there are a lot of amateur taxonomists out working and publishing on a regular basis. There was just a paper a couple months ago in PLOS talking about how many amateur entomologists there were, amateur taxonomists working in Europe, and the progress that the amateur community has been making there. But there's lots and lots of people, again, because there aren't that many jobs to become a pure taxonomist getting paid to do it. So many people use it as a hobby. It's an old hobby. Darwin was a taxonomist back in his day before he became an evolutionary biologist. Well, we're all evolutionary biologists, but before he became known for his work on taxonomy, before he came trying to postulate and share his ideas on evolution by natural selection, he established his academic street cred by becoming a taxonomist for barnacles. And so taxonomy has gone a long ways back as being an amateur or an unpaid hobbyist kind of discipline for a long time, and it's what's kept taxonomy going for a long time is amateur taxonomists. And so there's a bunch of resources, especially for entomology in North America in particular, where hobbyists can help contribute to our understanding of taxonomy, either by collecting and sending specimens. There's a bunch of different groups that are out there working with these citizen science groups where they ask people to collect ants in New York City, for example. There's the School of Ants project, and they're asking people in big cities to collect just the ants in their backyard. because we don't really understand it. And they found new things in Manhattan. They found a new species of ant in Manhattan a couple of years ago, just because nobody bothered to look. And so these citizen science groups are great. They also help to add specimens and photos to our records, so they can help us expand where we know these species are from. So if you visit bugguide.net, you can actually see all this user community where people are taking photos of insects, uploading them to this community, and then professional taxonomists, amateur taxonomists, and enthusiasts are all going through and identifying them. And now researchers are using these photos and these records to make new understandings of where these species occur in North America. So we've got amateurs that are contributing data. We've got amateurs that are contributing actual science, the taxonomy. They're undergoing revisions and describing species themselves. And then we've got amateurs that are just keen and want to know more about it. So taxonomy is, like I said, is very heavily driven by unemployed taxonomists in many regards, which it's fantastic in one regard, but it'd be nice for people to be getting paid and the academic acknowledgement for this work that they're doing.
That's true.
But lots of people love it, and lots of people do it as a hobby. So.
How has the revolution in molecular biology affected taxonomy? And the kind of work that you might be doing.
So, yeah, it's changed the game a little bit. Whereas the amateurs are great, they can do all of the morphological taxonomy they want. Molecular taxonomy is pretty much out of the hands of anybody that isn't funded by a major grant. It's expensive work. And so we've got molecular taxonomists are now starting to use more characters. So it's essentially all it is using DNA data as new characters to try and differentiate species and to create how they may be related to one another. And so this is something, like I said, that's not really in the reach of amateurs. And so this work is being done mostly by professionally employed taxonomists in research institutions like universities or government agencies to do the work. Again, it's mostly, at this point, just for figuring out relationships between species. There's been a little bit of work done on people trying to say we should be using molecular characters as the sole representation for species identification or differentiation, so describing the species based on genes alone. But so far the rules, I think, are rules for naming new species are still pretty strict on saying it's got to have at least some morphology in there as well. So while the genes and stuff are certainly contributing to a new avenue for taxonomists to explore, the citizen side of it is still very much a big portion of it. It's just two different tracks going on now, trying to look at high relationships and those that are still working and spending time on describing the new species rather than how they're all related to one another.
One of the areas I've been keeping an eye on is the advent of DIY bio and the lower costs of doing molecular biology these days, where we're getting down to $500, where you can kit out a home lab and be doing PCR and such like this. Do you see any potential future where the citizen science of taxonomy and the molecular biology, perhaps in the DIY range, come together?
I suspect that we'll probably see some people try it, but I think that most of the citizen science, the amateur taxonomists, are doing it because they like looking at bugs. And so I think they get most of their joy from going out in the field, collecting new insects, pinning new insects, keeping their own collections, having a physical representation of the phenotype, the actual, not just what the DNA, the string of DNA, but actually what the DNA creates. So bits and pieces of those DNA pieces come together. So I think at heart, most amateur taxonomists, and especially in my line of work, where I find the most joy is in the morphology, what the actual insect looks like and how it works. As an example, if we had just went off of DNA for this tiny fly, we wouldn't know anything about it other than it was a new species. By looking at what it looks like and comparing that to other things, we can figure out a little bit more about its biology. So that's why molecular taxonomy is probably never going to completely erase traditional morphological. But I would say that probably most amateurs will continue to do what they do, being expert naturalists, professional naturalists in that regard, and getting out there, finding new species, and not so much going towards molecular work unless they really need to in some regards. I could be completely wrong. Perhaps everyone's just itching to get their own do-it-yourself PCR and genome sequencing kit in their backyard, but I suspect that most of them are doing it for their love of the bugs rather than trying to get the DNA. But we'll see. It's going to be interesting.
I can see that. In some ways, the way you're describing it makes, the citizen science aspect of taxonomy sound a lot like amateur astronomy. People looking up at the stars just to go see them. Maybe not sitting at a $100 million observatory, but they've got a telescope and they like looking at the sky.
And they still make significant contributions to our knowledge of the stars, the same as amateur and unemployed taxonomists and naturalists and entomologists make a huge contribution to our knowledge in insect systematics. So,
I recall reading a story of a fellow. He was an amateur astronomer, and he was amazing at finding a particular class of celestial objects. I think it was supernovas, bagging them left and right, and making all of the multi-million-dollar astronomy teams look really bad. But is there any parallel in the taxonomy world? Are there superstar citizen scientists out there who are just bagging huge numbers of unexplained or undiscovered bugs?
I'm sure there are. Well, Ted McRae, he blogs at Beetles in the Bush. His day job is working for an agricultural company, but in his spare time he's a beetle taxonomist, and he's described several species. He's written keys. He's described many species. He's worked on the biology and the host biology of several groups of wood-boring beetles, and that's all as a hobby, essentially. He does that in the evenings and after work. He's done quite a few species. I don't know any numbers of the most prolific amateur entomologists, but I know that there are lots, and I know that there are some very prolific people out there who have devoted a lot of time to studying the species around them in many regards. So I can't give you a hard number on that, unfortunately, but there are a lot of people, and there are some very, very dedicated people that spend a lot of time building up their own personal collections and studying these things.
You mentioned Darwin and barnacles, and I know that Darwin was also a huge collector of insects, especially beetles. And one of the things I noted when I was reading about this was that he was engaged in what we would consider these days to be a social network. He had a whole group of people who he was corresponding with and trading specimens with and competing with, and it was this proto taxonomic social network. But it makes me wonder these days, what your feelings are on how the internet has changed the game, and what kind of effects it's had from your end and from the citizen end, and where that's headed in the future.
I think the internet is probably one of the single greatest tool of use for the taxonomic community. Like I said, it's connecting the scientists, whether they're employed or unemployed as taxonomists, with a huge user base of people interested in insects and taking photos. So there's been multiple cases that I know of where people have photographed insects for the first time in a new locality, so they've expanded range, they've shared their photos, sometimes new species, first time the specimen's been photographed, or collected online, and they share it on these forums saying, "What's this?" And everybody's like, "I don't know. I have no idea what that is, but that's amazing. Can you get more?" There's the increase in our knowledge of behavior and biology because of this it's been astounding. And so what it does is it more readily connects the amateurs in places where there aren't natural history museums to those people that are at these natural history museums, essentially. So most of the time you'll see taxonomists, especially insect taxonomists, centered around these big museums in Ottawa, in Washington, in New York, London, England, Paris, these sorts of places, because this is where we've got our giant data banks of hard specimens. So this is where we've been doing most of our work for the last 200 years. And so if you're in the middle of Alberta or Saskatchewan, you might not have ready access to these scientists. You might not even know that they would have existed back in the day. You would have seen a grasshopper, and you may have taken it to your agricultural rep, your local farm rep or whatever. They may have been able to help you, but if you had something that wasn't agriculturally related, it was pretty much out to lunch. So by the internet connecting and making available all of these professional taxonomists or expert amateurs to the greater community of people just walking through their backyard and finding a bug that they don't know what it is, it allows them to connect on a way that they have never been able to do before. And so far I've been talking about having the citizens, the general public, sending things to the researchers, but the internet's also helping us the other way around by allowing the researchers to contribute back and provide tools to the general public by the way of publications like identification keys. And so these tools, these identification keys, if they're done properly and if they're made open access and freely available, they allow anybody to go through and identify their bugs for themselves. So instead of taking that strange bug they found in their backyard, now they can maybe take it online and get an identification themselves, and then go to their respective professional and say, "Hey, look, I found this species. It's not known from this location, or I found it doing this. Is that known? How is that helpful? Is that cool?" And so these keys are becoming really popular, and again, I'm the technical editor for the Canadian Journal of Arthropod Identification, and the entire journal's purpose is publishing these keys open access. So the researchers are getting academic credit for this, not quite outreach because it's useful, but for publishing these keys and for disseminating their knowledge that they've acquired over their careers. And now interested naturalists and citizens and general populace can go out, use these keys, generally because they're well illustrated and trying to made by the magic of the web with as many photos and illustrations as we can cram into it for free. Hopefully they can identify and start contributing even more to our knowledge of these insects. So it's a two-way street for sure, with the general populace helping with biology and specimens, and then the group experts providing the tools for the general populace to understand and identify what they're seeing.
One more question on the technology front. I'm curious if the advent of mobile computing and like smartphones and mapping technology and GPS, what kind of effect has this had, and what do you expect in the future from that?
Yeah. So not only is it great because you can take these tools, so you can take your journal articles, like I said, your keys into the field with you if you saw an interesting bug and you wanted to identify it. You can do it right in the field now with your iPhone. But you can also use different tools like iNaturalist, which is an app where you can start recording your sightings. You can start using, grab my phone, I can't remember what the other one is. Project Noah is another one where these things are starting to aggregate, where you can start inputting your spottings, and then scientists can start using that data in their work. But also, the iPhone is the most popular camera. It's contributed the most photos to Flickr. So if you've got a camera with you at all times, it means that you can take a photo and increasingly better quality, even for macro work with your phone, and upload it to places like BugGuide or Flickr. So if you go to Flickr and you just search insect, you come up with, I can't remember what it was. I had the number last fall, and I can't remember what it is now. It was a huge number. I want to say either 150,000 or 1.5 million images of insects uploaded by users. I think it was 1.5 million, and about 150,000 of them or more had GPS data, so they actually were geo-referenced as well. And so this mobile technology of having the ability to take a picture and upload it to these various web databases while still standing in front of the insect is fantastic. And I think it's going to rapidly change the way that people are connecting with nature, because now they're no longer just saying, oh, there's an interesting bug. They can actually take a picture of it, potentially find out what the ID is, and then read about its biology on the Encyclopedia of Life, all within 5 minutes and still looking at the bug. So we come back to Star Trek a little bit here, and you get the little tricorder, and it comes up with beep, boop, beep, boop, beep, and it comes back with all the information in encyclopedia knowledge. That's where we're moving to, with the ability of the interconnected webs of information between taxonomy, naturalists, and this internet resources. So.
Funny you should say tricorder, since you could build your own now. Have you seen the plans for that?
No, I have not.
No, there's a fantastic guy out there. I'll put it in afterwards, but he's created what amounts to a tricorder, and it's got sensors.
Oh, sweet.
And I think it's based on an Arduino design, and it looks like one, and you can build it, and it's got touch screens, and it's awesome. But, it's funny because one of the big themes in molecular biology the last 20 years has been data and too much of it, right? Mm-hmm. Bioinformatics sprung up as a field largely because people needed to learn how to handle these huge amounts of data coming in. Mm-hmm. The way you're describing it sounds like taxonomy is on the verge of having a similar crush in terms of data coming down the pipe and needing new tools to deal with it.
No, I was just going to say, where are you dealing with that as we get into this whole genome as applied to relationships business? That's a big stop right now for a lot of the molecular biologists, dealing with these massive amounts of data and analyzing it and getting a tree of life out of it. It's just a massive amount of computing power. But
fair enough.
Dealing with the images and stuff like that, I don't know whether we'll have quite that overboard yet, but it's getting there.
Well, I have some ideas on that, actually. We should talk about that later. But
sounds good.
The one final question on this. What's the most interesting specimen you've ever come across?
The most interesting specimen I've ever come across. That's a good question. Let's see. Well, I mean, I've found a couple of specimens, new species that are currently awaiting description. I'll be describing them later this summer. So through the course of my master's work, I found several new species in that regard. Just this couple of weeks ago, I was up in Ottawa and I found a species that I thought was for sure in my group, but it's not. It's a new species, probably from a completely unrelated group of flies, and it just looks almost exactly, except odd, like my group of flies. So totally unrelated, but the shape and the size of it and the color patterns and stuff like that completely mimic my group. So that was pretty sad as well. But I don't have any weird, tiny, super tiny flies in mine. Mine are all different colored and big.
Well, it's been it's been well over an hour at this point. I didn't intend to turn it into interview you episode, but that's kind of the way
next time I'll take a back seat and I'll interview everybody else.
Well, actually, no. I really actually want to thank you for that, because it was an interesting insight into taxonomy, which I've never had before, and I feel like I learned a lot about it just from listening to you talk about it. You talk about it very well.
Thanks.
So, but I think at this point we can pretty much wrap it up and call that the third episode, since it's now 1:30 in the morning, and I would like to get something resembling sleep today.
That sounds like a good plan to me.
So that's episode 3 of the podcast. For anybody who happens to be listening to this, if you'd like to comment or ask questions or otherwise be involved in the conversation, you can drop us a line at breakingbio@gmail.com or follow us on Twitter at breakingbio. And I'd like to thank Morgan for joining today and for turning unwittingly into the subject of the podcast. And we'll see you next time.